From Microbe TV, this is TWiP, This Week in Parasitism, episode 141, recorded on October 30th, 2017. Joining me today, here in New York City, Dickson Despommier.
Hello, Vincent.
And from a remote location, Daniel Griffin.
Hello, everybody.
Hey, Daniel.
I feel discombobulated not doing a billboard, you know. Our ad contract is up. It's finished.
Really?
Yep.
Oh, my.
That's it. Done.
Wow.
And we
post your bills here?
We have fed the world, and that's it.
That's the end. Oh, too bad.
Yep. No more.
Oh, well. There's something else out there, I'll bet.
Let us know if there's something else out there.
I just might.
So we can go right into the weather.
Right.
It's a beautiful day.
No, the case. We can go into the case. We're skipping the weather. This is TWiP.
That's right.
That's right. This is not TWiP. It's almost sunset, Daniel, by the way. Do you see the sun setting? Oh, you're in a room with no windows.
No. But I just barely finished my last consult of the day, so the sun was out as I...
All right.
Right.
Daniel, remind us of the case from TWiP 140.
I will. You know, one of the things I was thinking before I started is, our listeners, when I listen to the podcast after the fact, which I do, I get to hear that really nice mood music at the beginning. I almost wonder if we couldn't get us sort of in the mood for our case. But okay,
case, a song of a case.
So this is actually, hopefully this is something we'll see more of. This is a case that was sent to us by one of our listeners. This is Dr. Arthur Mamello, and he is a physician in northern Kenya. He's actually moved around to a few different places in Africa where he's been in practice. But this is a case he saw recently, and it's a 1-year-old girl, and she was brought in by her mother with skin lesions that had developed 1 week prior. There were 5 lesions in total. So let's go through. 1 on the forehead, 1 on the neck, 1 on the back, 1 on the chest, and then 1 on the right arm. And I should mention, we actually have video of the 1 on the right arm. The lesions look like boils or furuncles, we describe them. So they're sort of raised, they feel fluctuant, but they are changing in size, appearance, and there was this description, which I think Dickson felt like we were giving too much away, as though there was like something was actually moving under the skin. My comment was, I'm not sure how we got that history from 1-year-old, but maybe the mom felt like there was something moving under the skin. They are described as painful, and the child was scratching them. The child is currently breastfeeding and is breastfeeding well. No other complaints. The child was born at the Nyahururu County Referral Hospital and had gotten all the vaccinations at the Melwa Health Center, it's a rural health center. All the vaccinations are up to date. The family lives in a wooden house with a dirt floor that is roofed with corrugated iron sheets. It's actually quite common in many parts of the world. This dwelling has 2 rooms, and they do not sleep on the dirt. They actually have raised platforms, raised beds where they're able to sleep. There's a big community dam in the neighborhood with stagnant water throughout the year. They don't use mosquito nets. They have reliable, clean water that is supplied from the government. They have 1 dog, but there's also a neighbor's dog that visits quite often. They hang their clothes on the clothesline after washing. They never dry their clothes in the grass, and the clothes are not hot ironed. On examination, the child is breastfeeding well, no fever, no pallor, no jaundice, appears not in distress. There is some enlargement of the lymph nodes in the back of the neck, so I'll say occipital lymphadenopathy. These are tender and mobile. There are these lesions, which are described as furuncular on the 5 places mentioned. They're about 1 to 3 centimeters in diameter, so it's about an inch, and about 0.5 centimeters up, so we'll say a quarter or a third of an inch raised. They are tender, and they have this central punctum from which there's a sort of a light reddish clear fluid that is draining. This is a rural health center, so the only labs done are a peripheral blood smear, which showed increased eosinophils, neutrophils, and there's an HIV test, which we learn is negative.
And I guess we also have to add that it didn't show any malaria parasites either, right?
At least not that are reported, right?
Exactly. But that's what the thin smear was all about, no?
Yeah, I guess we'll comment on that. So when you get a smear, you can do your thick and thin smear.
Yeah.
And, I think the more we do this, the more it helps to refresh people. Your thick smear is a really very sensitive screening technique for is there or are there malaria parasites in the blood. Your thin smear is going to have lower sensitivity, but it's going to allow a visual distinction between different types of malaria, let's say vivax, falciparum, etc.
Exactly.
And some other, I think I remember on some tests, like what parasites could be seen or what infectious disease could be diagnosed on a peripheral blood smear.
There you go.
Anyone listening wants to respond to that. That's one of those.
Oh, that's a good question, actually.
Yeah, because there's ones that we quickly think of, and then there's a couple less common.
This is true.
So non-parasitic actually are the ones that people don't remember.
Well, we had quite a few responses here.
We did. First of all, it means that people are listening. This is good. This is very good.
Chris writes, Dear Twipsters, the season is finally changing in Athens, Georgia, where it's a very tolerable 73 F, 23 C, humidity 32%.
Nice.
I just listened to the latest TWiP, and there's no way I'm going to miss sending in a case diagnosis this time.
Okay.
Chris was the first one, and he sent his diagnosis for the previous case the day after we released it.
Cool.
Which means he was late.
Well.
The presence of furuncles and serosanguineous fluid of a 1-year-old patient paired with her location in Kenya make me think immediately of a fly infestation, particularly of the tumbu fly, Cortylobia anthropophaga. The presence of a central punctum on the furuncle. What a great phrase. May be enough to suspect myiasis, after which the larvae may be persuaded to emerge by cutting off their air supply. The larva's posterior spiracles require access to outside air for respiration and can be covered with a layer of petroleum jelly to induce suffocation.
Indeed.
I was unable to find reports on how long this process can take, so the disposition of the patient may be of special relevance. Once partially emerged, the larvae may be removed with forceps or simply squeezed out. Care should be taken not to rupture the larvae in situ for all the usual reasons. Secondary bacterial infection is uncommon due to the antimicrobial products of the larva, but an antibiotic regimen is still recommended for safety. At this point, definitive species identification can be made by examining the larva. The number and location of spines, as well as overall shape and size, can be diagnostic. 3 Cortylobia species are known to infest humans: Anthropophaga, Rhodini, and Rhodende. The first is by far the most common, and considering geography, the most likely diagnosis. Removed larvae can also be allowed to develop in the species, more easily determined by features of the adult fly. While potentially fascinating from an epidemiological perspective, all of this is likely dubious interest to the patient and her mother. You notice how fast I'm reading?
I do.
You're like on 1.5x. This is the way I listen to your virology lectures.
Have another cup of coffee, Vincent.
Now, I haven't had one all day. Now, for the source of this infestation, if left unperturbed at 8 to 12 days post-infection, the larva will have matured to the prepupal stage. From there, it will drop free of the host and abscond into the soil to continue pupation until it emerges as an adult fly. Females live for about 2 weeks and lay their eggs in batches of 1 to 300 in sandy soil, typically contaminated with feces or urine. However, if damp clothing is available, as in this case, the flies will readily lay their eggs on this. After a few days, the larvae hatch and penetrate intact skin while the clothing is being worn. Because of their thinner skin, children more commonly become hosts. Dogs serve as a reservoir for the fly, which are reported to be present around the home, and the patient's mother may be advised to examine their pet for signs of infestation. Ironing clothes will kill eggs and may be used to prevent future infestation, but I know from personal experience how hard it is to keep up with ironing. Very interesting case. I look forward to your insight. Stay fly. Chris.
There you go.
Dickson.
Wink writes, Dear TWiP professors, a general gender-neutral term. The poor child in your most recent case sounded, from my Western Hemisphere orientation, like the botfly. I didn't think that species was in East Africa, so I turned to the textbook and read myiasis caused by Cortylobia anthropophaga. The tumbu fly is a larval parasite of humans and other animals, especially rats in Africa. This made me concerned that the case history was implying that this infant was spending time near rats. I guess it is important to be reminded about the lifestyle of those left less fortunate. So that is my guess. My eyes is caused by the tumbu fly larvae. Wink from Atlanta. Thank you, Wink.
Daniel.
John writes, Dear Doctors TWiP, my guess for the case of the 1-year-old girl with itchy upper body lesions in TWiP 140 is onchocerciasis, more commonly known as African river blindness, though it is not restricted to Africa. More on that later. The disease is caused by the nematode Onchocerca volvulus and is spread by the black fly, Simulium. These flies rely on fast-moving rivers to breed, hence the common name of the disease. The range of the flies is several miles from its breeding grounds, and in the past has meant that large regions around rivers were not inhabited. The disease is endemic across sub-Saharan Africa and parts of northern South America. Humans are the definitive host. When a human is infected by a larvae from a female black fly bite, the larvae migrate to the subcutaneous tissues and mature into nodules, which vary in size from barely discernible, 5 centimeters in diameter, very discernible to 5 centimeters in diameter.
Right.
Maturation takes 6 to 18 months. When mature, the males and females mate, and the females produce about 700 microfilaria per day for their 8 to 10-year lifespan. The microfilaria migrate throughout the skin and eyes. Subsequent bites from black flies pick up the microfilaria, which mature into larvae in the flies and migrate to the proboscis of the fly to continue the cycle during the next blood meal. When alive, microfilaria are non-immunogenic. Interactions between antigens from dead or dying microfilaria, their endosymbiont Wolbachia, and our immune system are thought to cause the lesions that lead to the various symptoms in the eyes, including eventually blindness, which may take 20 to 30 years to develop. The antigens also cause an increase in neutrophils and eosinophils. The skin symptoms, severe itching, thickening, depigmentation, are mainly caused by antigens from the microfilaria alone. The location of subcutaneous nodules on the body of the infected person depends on the biting pattern of the flies and the clothing styles of the inhabitants of the region. Typically in Africa, the nodules are on the lower body, while in South America, the nodules tend to be on the upper body. The patient in this case is a 1-year-old girl, so styles of clothing for babies may account for the difference from the usual African pattern. Diagnosis can be made by microscopic examination of 2 to 5 millimeter square bloodless skin snips from a region thought to have a high level of microfilaria. Multiple snips should be examined to increase sensitivity. Alternatively, the Mazzotti test can be performed. And from Parasitic Diseases 6th edition, we have a quotation here. The Mazzotti test is a provocative challenge test using a 50 milligram dose of diethylcarbamazine. Within 3 hours after treatment, patients with O. volvulus infection will develop pruritus. In heavily infected patients, the Mazzotti reaction can be severe and may exacerbate the ocular pathology in a patient. As an alternative, some physicians perform a patch test by applying DEC to a small region in order to elicit a local Mazzotti-like reaction. Treatment is typically with ivermectin. Ivermectin binds to glutamate-gated chloride channels in the microfilaria, resulting in paralysis and death of the parasite directly or by starvation. The adults in the nodules are unaffected, and treatment must be repeated at 6-month intervals while the adults survive. The nodules containing the adults may be surgically removed. The death of the microfilaria as a result of ivermectin treatment may trigger a severe immune response in the patient. This is particularly true in patients co-infected with loa loa. Patients at risk of co-infection should be checked. It is worth noting that Merck, the manufacturers of ivermectin, pledged in 1987 to donate the drug to anyone who needed it for as long as it was needed. I think I've plagiarized, referenced your book enough for one email. Thanks to all for the continued spread of information and education. Regards, John in Limerick, where the recent weather has included ex-Hurricane Ophelia and Atlantic Storm Brian. Mm-hmm. Yeah, I think that was unique for them, right, to get hit by a hurricane. So should we say right off it's not onchocerca, but talk a little bit about river blindness? What do you think, Dickson? Are you thinking onchocerca volvulus here?
No.
Yeah, and it's interesting though, because I like the fact that he thought about it when you see all these nodules. But I think there were certain characteristics, we'll call it the breathing hole, the feeling of
exactly.
I think a movement in there. Yeah, these would not be, but you can actually, and I think also the geography, right?
Correct.
We're in Kenya,
That's right.
so we're really outside of the zone where we're going to see.
100% right. But it was a very nice plagiaristic trip through Onchocerca from our chapter, and I like the way he presented the data. So it's worth a note in passing, but I don't think that this is going to win the first prize on this one.
Yeah. But no, I liked it. It's always, it's one of those.
No, it was a good review.
Acidic diseases that we enjoy. So
nice
thanks
review. So the couple of features that you would have pointed out anyway is that the lesions from Onchocerca don't really ooze, and there's no central punctum. So punctatum, I guess, is the way to put it. So, I mean, you could distinguish these by looking, but we didn't show you pictures. So that's probably why you might have confused them.
Yeah, but it is true. If you do surgically remove these, it has been shown to actually be beneficial in treatment.
Oh, yes, no question.
Yeah. So and I think the nice thing of nowadays, most people are not doing the full-fledged Mazzati test. They're actually going to apply a small amount, and that's going to. But we also have better access, I think, to microscopic testing.
True.
So, all right.
All right. Alan writes, "Dear TWiP Wataalamu, warm Kona greetings for anyone who has lived any time in rural, central, or east Sub-Saharan Africa. The patient history described by Dr. Mumelo screams mango fly or tumbu fly, Cordylobia anthropophaga, particularly the described dirt floor nearby dogs, and clothesline drying without ironing. See anthropophagy with its apt Greek name, human eater, causes cutaneous myiasis. Each female mango or tumbu fly lays 100 to 300 eggs in contaminated soil or on damp clothing or bed linens, often drying after a wash. Unless ironed, the eggs on the clothing or linen will hatch into larvae after 2 to 3 days and penetrate the skin. With larvae that hatch in soil, they can remain viable in soil for 9 to 15 days, and any disturbance of the soil causes them to wriggle to the surface to penetrate the skin of the host. At the site of penetration, a red papule forms and slowly enlarges, first with only intermittent itching, but pain develops and increases in frequency and intensity as the lesion becomes a furuncle. The furuncle's aperture opens, permitting fluids containing blood and waste products of the maggot to drain, as described in the patient history. After 8 to 12 days, the larvae has gone through 3 larval stages before it reaches the prepupal stage, at which point it drops to the ground, buries itself, and pupates into an adult fly, able to reproduce and begin the cycle again. Humans are, in fact, accidental hosts. Dogs are the most common domestic host, and several species of wild rats are the preferred field hosts. Treatment is simple. Cover the punctum with petroleum jelly to cut off the larva's air supply, which forces it to the surface where it can be captured with forceps. Sometimes the punctum needs to be enlarged a bit, but the furuncles are painful, so prevention is much preferred. Iron the clothes. Okay, due diligence. Here are some other possible causes. According to the Inestimable Parasitic Diseases 6th edition, Wolfartia and Sarcophaga can also cause cutaneous myiasis in unbroken skin, but these flies don't live in Africa. The Congo floor maggot, Ochmeromia luteola, could be the cause if the patient slept on the floor. The history said they slept on beds, so this is unlikely. Chrysomia, the old world screw worm, as well as green or blue bottle flies, Lucilia or Califera, can cause myiasis, but these flies only lay eggs in wounds or mucous membranes, and the lesions described were of an unbroken, intact skin. So I'm sticking with the mango or tumbu fly as the most likely cause. Thanks for another great TWiP case study, Alan Robbins, Kona, Hawaii. Wasn't there an example once on TWiP of one in which the fly emerged from the skin? It didn't develop in the soil, right? The Super Bowl party, right?
So no, but that was the bot fly on the butt from Belize. But that was a New World bot fly instead of a mango or tumbo fly, and so it's actually the larval stage that's
the Dermatobia hominis.
Got it.
And I think we may have also done Tunga penetrans, which is a flea, and you actually have the female flea in there, and then you've actually got
That's right.
Dickson, you're next.
Okay, Ryan writes, "Hello, doctors. The case presented in episode 140 sounds to be caused by myiasis-causing dipteran fly larvae. The description of a furuncle boil is characteristic of larvae that have taken residence under this poor girl's skin in the 5 locations described. I believe the girl acquired the infection from eggs deposited by the adult fly on her clothes while they were drying. It is also possible the girl was exposed to the eggs by fecally contaminated ground caused by the dogs. The warmth of her body would excite the eggs to hatch and penetrate her skin. The larvae are developing under her skin, hence the central punctate area to allow for waste discharge and gas exchange required by the parasite. A quick Google search finds cases of these infections cause eosinophilia and enlarged lymph nodes, which fits this case well. Painful, itchy lesions are expected to worsen as the larvae grow, but eventually the larvae would make its way out of the host, drop to the ground, and continue its life cycle to become an adult fly. Reading Parasitic Diseases, 6th edition, I believe the culprit to be the tumbu fly, Cortylobia anthropophaga, due to the geographic location of the patient. The species appears to be the only cutaneous myiasis-causing larvae in the region. In the same region, Chrysomya species infect wounds or mucous membranes, which were not included in the presentation. Lucilia and Califora species prefer to feed on necrotic tissues, so they're good at debriding wounds, basically, and that's what that means. The Congo floor maggot, that's misspelled here, but I think it's A. luteola, does not seem a viable culprit because they do not penetrate tissues. Cuterebrus species in the USA or Dermatobia hominis in South and Central America may have caused similar lesions to the ones presented. To treat this infection, petroleum jelly can be used to cover the lesion to starve the larvae of oxygen, forcing it to the skin surface. The larvae can then be surgically removed. Thanks again for the work you put into this educational and entertaining podcast. I particularly enjoyed the Blastocystis paper discussed in the last episode. A friend of mine was one of the unlucky few who appeared to harbor one of those pathogenic variants. As of August this year, he was told he was Blastocystis free. Well, that's good to know. All the best, Ryan.
Daniel.
Hannah writes, Dear TWiP doctors, as soon as you mentioned unironed clothes, I knew what it was.
Excellent.
Cortylobia anthropophaga, the mango or tumbu fly. The mango fly, not to be confused with the mango-loving flies in a previous TWiP episode, is a common cause of myiasis in Central and Eastern Africa, including Kenya. The adult fly lays a large number of eggs in urine, feces, contaminated sandy soil, or, as likely happened in this case, on damp clothes or linens hanging to dry. There, the larvae hatch and wait for a host, a human, for example, who didn't iron his or her clothing. It then burrows into the skin and a furuncle forms around it, becoming more itchy and painful as the larva grows. Meanwhile, blood and larval waste products drain out of a hole. Eventually, the larva leaves the host, pupates in the soil, and develops into an adult fly. All of this can be avoided if you iron your clothes before putting them on. The only reason I know anything about mango flies is this Washington Post article that I read last month. It's about the parasitic horrors faced by wildlife biologists, and it features a few other TWiP favorites as well. And we have a link here.
Nice.
Want to be a wildlife biologist? Beware the eyeball leeches and chest maggots. Washington Post. And that was back in September 22nd.
Cool.
I can't think of any other East African arthropods that would present with similar symptoms. The closest would be the human botfly, Dermatobia hominis, but it's not present in Africa. It's native to Central and South America, and the description of the lesion doesn't sound quite right. Tunga penetrans is present in Kenya, but they tend to end up embedded in feet or hands, whereas this child has lesions all over the body. As for non-arthropod parasites, the lesion's wrong for leishmaniasis, wrong shape, plus leishmaniasis lesions don't move, and doesn't match any worm that I know of either. If it weren't for the changing shape and movement, my money would have been on some routine bacterial infection. All the best, Hannah.
All right, we have two more. Yosef writes, Dear Triple TWiP. Wow, that's a bit of a tongue twister. I'm not too sure of my diagnosis, but the differential that I'm thinking of are mainly Trypanosoma brucei gambiense or cutaneous leishmaniasis. Child has occipital lymphadenopathy, which could be Winterbottom sign, which is associated with West African TWiP. There is usually just a single chancre at the area of the black fly blight, and it shouldn't be changing in size or looking as if there's a worm underneath. There is usually also no eosinophilia noted with the infection. Regards to cutaneous leishmaniasis, the skin findings are also usually different. The initial lesion often swells, but then starts to ulcerate, which was not mentioned with the case. There is also peripheral eosinophilia within the cutaneous lesion, but I don't think there is usually significant increase in the serum eosinophil concentration. The last diagnosis that I was entertaining was guinea worm due to the mention of worms moving within the skin lesions, but Kenya has not had guinea worm infections in many years, and unless this case was sometime in the past, it cannot fit. Furthermore, there's a 1-year-old. Why would they be drinking water at this point? Lastly, it was mentioned that the family had access to clean water. Overall, I am not sure of the diagnosis, and I'm not confident of my thoughts. I'm looking forward
in any of my thoughts. I like that.
Well, I'm looking forward to the answers. Sincerely, Yosef at the Donald and Barbara Zucker School of Medicine at Hofstra Northwell. Well, the school changed its name.
Indeed.
You know, I think it's very brave of Yosef to write and not having a good guess. Most people would not, I think.
That's true.
That's how you learn, right?
This is true. Well, another way to learn, of course, is to go look these things up in a book.
I'm sure he did.
Okay.
Don't you think? Or is he trying to pull it out of his brain? I don't know.
You know what I was going to say? This is actually interesting. When I was doing my infectious disease fellowship training, and here I was, with many really bright individuals, cutaneous myiasis is something that is really not part of the standard training.
That's true.
And so unless you know about this, you start thinking about all these other things of which you do know, and you say, let's think, I'm in Africa, I've got these nodules, and you might think of, Winterbottom sign. I mean, that shows a level of education and learning. You think of onchocerciasis nodules, people brought up cutaneous leishmaniasis.
That's right.
But once you, as we will perhaps realize, once you're aware of what is causing this and the clues, the features of the case, then it becomes a recognition, I think. You say, this obviously fits that bill more than anything else. But I applaud that too, because, the best way to learn, I think, and it's maybe frowned a little bit on because of the kindler, gentler approach to learning these days, is you put yourself out there. And then you say, this is what I'm thinking, basically, because we don't know what to teach until we know, what sort of deficiency is there. And then you say, okay, I see that you're thinking this, but let's work through and get you to a higher level.
Right.
Last one, Dickson.
Okay. Gavin writes, Dear TWiP team, I believe that the child in TWiP 140 is suffering from a case of myiasis caused by the tumbu or mango fly, genus Cortolobia. This is consistent with the presentation of furuncles with a central punctatum and the presence of seropiginous fluid. Serosanguinous. Sorry, I'll repeat this. Presence of serosanguinous fluid, eosinophilia, and geographic location. I'm running a little behind, so I don't have time to look up the difference between C. anthropophagia and C. rhodhani, but I feel that we can safely rule out D. hominis, which is the botfly, and Cuterebra, which are more common in the Americas. Wolforthia species are located around the Mediterranean basin and can also be ruled out. My differential includes insect bites, pyoderma, cysts, and tongiasis. Diagnosis can be made on clinical grounds or with the aid of an ultrasound. Maggots can be expressed with a kitchen tool, which one? Or encouraged to leave by, hopefully not a pizza cutter, or encouraged to leave by smothering with paraffin. Oh, that's an interesting take on that. And paraffin would work, I'm sure. Cheers, Gavin.
Okay. Nice response this time.
We got a lot of nice responses, is right. And even the ones that were wrong were complete in terms of what they knew about that, except I must take some exceptions with Yosef's answers for trypanosomiasis, blaming the bite wound on a black fly, because that was actually a tsetse fly. I would have gently corrected him.
Yeah.
It's okay. No, it's okay, because when all this stuff is floating around in your head and you're new at it, you remember bits and pieces from each of these parasitic life cycles. And unless you've got a Virgil to lead you through the forest of confusion, so to speak, then you're likely to end up with that.
At that school, they all have your book.
That's true. That's true.
They have the physical book.
This is right. I agree with you.
You know what they're lacking? They're lacking a course.
Guru.
A course, right?
Mm-hmm.
So you get your book, but you've got to go through it. And I try to throw, I think, mnemonics to help people remember. So the black fly is going to give you river blindness, right?
Yep.
The deer fly is going to give you loa loa.
Yeah.
And then I think we could just remember the tsetse fly is really cool, basically live birth, and so that's going to give you these very interesting sleeping sickness things. You can dream about this wonderful, amazing fly in your sleep,
which you can now cure with a single pill.
Yeah, we need to talk about that in the future.
I'd like to.
I did want to jump in on our last. So our last writer made some interesting comments about the difference between C. anthropophagia and C. Rhodani. Yeah, which is hard for me to pronounce.
It's a place in South America, I think. That's right.
And so the interesting thing, how can you tell? Is there something about this feature that would help you? So the common name for Rhodani is actually Lund's fly, and Lund's fly deposits its egg in the soil. So when we see those, they tend to be on the feet, so they tend to penetrate the skin of barefoot walking people.
Interesting.
So this person, I would say, this 1-year-old breastfed, we would think actually more of the mango fly, I'll say, when we get to it, the tumbo fly,
right.
Because of the location of these. So but that's interesting. He brings them to up because they're both in this area.
Okay, they are both in the area then.
Yeah, they're both in the area. But the issue would be, the location sort of helping, because I think as we'll go on, we'll talk about, can we identify the actual maggot larva from looking at the images that we were sent. But I think one thing interesting about this case is everyone, including you, Dickson, it was unveiled to you before we even presented this, right? So you already have all the information.
Yeah, I'm afraid so.
Actually, what we read last time was pretty much everything, right?
Yeah, we did.
We left a few things out, right?
We left the videos. We didn't show the videos.
Yeah.
I think I made a few tiny little changes just to make it a little bit more of a challenge.
Right.
So, Daniel, if you were the physician in this case, examining the child, what would have clinched it for you?
Well, I think the actual visual, seeing these breathing holes, the punctum in the center of each one of these, and then actually the whole description of something moving, and then as we go on to see in what was sent to us, when pressure was applied, you actually had a maggot erupt out of these different sites. And that was pretty compelling.
So you want to be a doctor, do you? Here's what you have to do.
We had audio, actually, so the child was not happy. He was not enjoying.
No, I don't blame him. That's a interaction with to go through
medical patient.
Yeah, doctors sometimes cause pain to children.
They do when they don't want to, but they know you have to in order to move on to the next step. Even
some adults yell.
Oh, tell me about it. They get the sight of a needle and they start to cry.
Yeah.
It's true.
Have you heard of the latest podcast parasite?
Have not.
They crawl into headphones lying on the table, and when you put them on, they crawl out of the headphone into your ear, and they make you say dumb things.
Get out of here.
A podcast parasite.
It's
a newly emerging phenomenon.
I probably suffer from that disease. Is there a cure, doctor?
I just thought of it. Stop podcasting. And then they crawl out.
It reminds me of a scene out of a Star Trek movie where they had a little worm that goes into your head, right?
Is that right?
Remember that one?
I don't.
So wasn't that Wrath of Khan?
The Wrath of Khan.
The Wrath of Khan.
Exactly right.
Isn't that what
With Ricardo Montalban.
What do you call those worms? The earwigs. Don't they crawl in your ear?
Earwigs? No. No.
I know. But they sound like they could, right?
They sound like they could. That's true.
Is there an earwig in parasitic diseases?
No. Not really.
Are earwigs harmful to humans?
No. They're not.
They do not.
They're one of the oldest orders of insects, though. They've been around a long time.
Really?
Yep.
I like using the word order.
Indeed.
Because as you know, it's also a word for a religious
Could
thing,
be.
right?
That's right.
One of the oldest orders of insects. I just see the insects praying.
All in a row with their little robes, perhaps.
Well, any listener who's interested, you could send us your case history. That was very good.
Absolutely.
Of the good doctor there. So that was fun.
Yeah.
Gave Daniel a week off, right?
It's fantastic.
Yeah, should we talk a little? Let's talk a little bit more about it because people brought up, and there's this, I think, a very interesting distinction between the type of cutaneous furuncular myiasis that we're seeing here from Africa and the cutaneous furuncular myiasis that we see in what they call the New World, which is getting a little old, but and one of the big things I think is an exciting difference is this issue of pheresis. Right, that's a fancy word for something that the human botfly does, which I think gets people very excited. So what the human botfly does itself, it sort of like steps back and it actually puts its eggs on other insects, and then those blood-sucking insects alight. Is that the right word? They alight on a human being or any mammal. It doesn't necessarily. I actually think humans are probably not a great target, but they alight on when they alight on a human, the eggs will actually, there's an emergence, and then that's where you get the penetration. And I think we were talking about our guy in from Belize, and how did it sort of get into that buttock area where the pheresis is not the way our mango flies. What our mango flies will often do is they're actually gonna deposit the eggs on the wet clothing that is hanging up. And actually, when I made my daughter Daisy listen to this, and I sort of made a little comment. I was like, "Oh my gosh, they didn't throw the clothes in the dryer or iron them." And she's like, "I know what that is." When we were in Africa visiting her cousins, they made a point of, don't not hang your clothes, out on the line. Right. And what ends up happening here is when you put those clothes on your body, then the warmth of being in your body triggers the larvae to penetrate.
Right.
So actually the location, that's why you get all these different locations, because you put the shirt on or whatever bit of clothing.
So Bob Grodz, when he gave this lecture here for many, many, many years, and we should pay homage to his contributions, used to also indicate that because the tumbu fly is attracted to urine and feces, and that's usually in the ground where most animal defecations exist, that's where the eggs are deposited. But in diapers that are not cleaned properly and then just hung up to dry, say they're washed in contaminated water to begin with, right? And there's no soap. You're just going to rinse them out and then hang them on the line. There's still some residuum of odor from the feces and urine that attracts those flies to those pieces of clothing. I think if they were perfectly clean, they wouldn't, because I think it's the chemical trail that the fly follows to the urine and feces, because it represents an energy source for the newly hatched larva. The feces does, at least.
I think in general, I think that is probably true, because you see this more when people they would lay a blanket on the ground to let it dry.
Oh yeah.
And then there's a dirt contact, and then the dirt I think provides some. But it's really interesting, the signal that somehow the coming in contact with the warm body triggers the emergence and the penetration.
Absolutely, those are called environmental cues. For those of you out there that want a name for that. Or tropisms, and these tropisms, this is a thermal tropism that we're talking about now. Yeah. Vincent is making interesting machinations. Questing. He's questing with his fingers like he's a bunch of larvae.
Boy. What's on the grass that quest when you walk by?
Those are the hookworm or strongyloides larvae.
I learned that from your lectures.
They come they almost look like flames, slow-moving flames when they work together. There could be hundreds in these bundles. And they're quite interesting to watch.
I saw a movie at a presentation on Saturday of a malarial parasite penetrating a red blood cell.
Look at that.
Oh, man, the thing was wriggling around.
Oh, yes, absolutely.
Trying to get in. It was so monstrous.
Vigorous. Exactly right.
Speaking of malaria, we do have a malaria paper today.
We do have a malaria.
Daniel, anything else you wanted to say?
No, I'll just finish this off quickly, just so we've got our myiasis understood. So we spoke this time about a cutaneous furuncular myiasis, but there also are a few other forms, right? There's the wound, myiasis, where they actually get into wounds, and sometimes that might be good. That might not be so bad. We had a case of, we call an accidental myiasis, which wasn't really even parasitic, where someone had sort of swallowed and the mango loving larvae came out in the stool.
Right.
Really doing nothing other than maybe having an aesthetic negative impact on the human being and getting transported to some other locale. Occasionally they can get into organs. And so there's a few other forms of myiasis, and it's unfortunately something that I don't think we maybe learn about. And so I think that was the challenge, that Joseph, his education is working against him. His head is full of all kinds of molecular biology and other things. And, anyway, but yeah, I hope everyone enjoyed that. And thank you for sending in a case. Hopefully we'll get some more cases sent in.
Just as a passing note here, Daniel, have you ever observed physicians using maggots to debride wounds?
I think that we talked about this before. When I was at Bellevue, what we would do is leave them on to some degree,
right?
To let them. But there is an issue. There was a belief, which we now realize is not 100% true. The belief was that the maggots would only eat dead necrotic tissue, but they'll keep going a little bit. So so you want to be somewhat careful.
Dickson, is it true that museums use bugs to remove the tissue from bones?
They do.
What sorts of bugs?
They use beetles.
Beetles.
That's right.
The fabulous four?
-1.
Okay.
That's true. They do, though. They use the beetles to do that.
They're very efficient, right?
They're extremely efficient. Yeah,
wow.
That's right.
All right. We have a paper from Science.
We do.
That Dickson found.
I actually have the paper.
He's got the actual paper. He ripped it out of his...
Here, that's a real piece of paper here.
Driving mosquito refractoriness to Plasmodium falciparum with engineered symbiotic bacteria.
Right.
The first author is Xibao Wang, and the last author is Marcelo Jacobs Lorena.
Yes.
From the Chinese Academy of Sciences, which is in Shanghai, and the Johns Hopkins Bloomberg School of Public Health.
Indeed. Now, Vincent, before we get into this paper, you've read this paper, and you like this paper,
I like
too.
it a lot, yeah.
And how many TWiP or TWiX shows do you host?
Currently, virology, parasitism, microbiology, evolution, urban agriculture. Does that still count?
Well, maybe.
And then this week, we're releasing the first immune.
Okay.
6.
Okay.
So I'm out of control.
Right.
So is that where you're bringing me here?
No, I'm not. I'm asking you in this paper.
Now, how many are combined?
How many TWiX papers?
So we have malaria. So that's TWiP.
That's right.
We have bacteria. That's TWIM.
We do. And it's genetically engineered with a virus.
Where did you get the virus?
Well, you have to put the genome inside of the microbe, don't you?
Yeah, but that's just by transformation.
Oh.
I don't think we have a trifecta here.
That's too bad.
It would have been nice.
No?
No, I don't think they used any virus.
They didn't use phage to get inside?
No, they just transformed the bacteria.
Oh, darn.
All right. So this is cool because the idea here, they say, hey, don't kill the mosquitoes, but convert them to an ineffective vector of malaria.
There you go.
And their idea is to target the midgut stages.
Right.
Now, I didn't realize this, Daniel and Dickson. Oh, 2 Ds. How about that? Malaria parasites suffer losses during their development in the vector, with the most severe bottleneck of Plasmodium in the lumen of the midgut. Didn't know this. Did you?
I had heard that before from Bob Gwadz, who actually studied malaria in mosquitoes. Yes, I had heard this.
Obviously, it doesn't stop them.
No, but it limits the number, I think.
Otherwise, it would be worse. You deliver more.
Some mosquitoes can be killed by too much malaria. That's true.
So they go ahead, Aunt Daniel. What were you going to say?
I was gonna jump in. It's an interesting issue, and when I read a little bit more about this issue, I was kind of intrigued. Right, I never really, we. As physicians, we focus so much on the patients, on the human cycle.
Yep. And
even when we look at the chapter, here's a chapter on malaria, and we see, you get bit by the mosquito, the liver, you go through this whole cycle. And then there's like, that's what's going on in the mosquito, kind of ignore it and walk away. But there is this really interesting, understood, studied parasite cycle in the mosquito vector.
Yes.
And The first thing I think I was shocked when I looked about it is the amount of gametocytes that a mosquito can obtain from a blood meal.
Indeed.
And so these are kind of the numbers to give orders of magnitude to this. So a mosquito can ingest, and this still shocks me, but this is what's in the literature, 10,000 gametocytes from a meal, right? That's absurd. That's, but that's what they say.
And half are male and half are female, presumably.
Yes, I think it's 51%.
So
no, I'm joking.
Yeah, I mean, I just thought I'd throw that in.
It is a mix, right? And then, so then they come together, so they fuse, right, form a zygote. And now we've got half roughly, but that process is inefficient to the point where you lose an order of magnitude. You go from 10,000 to 1,000.
That's true.
But then when you go from these kinases to the oocysts, you get down to less than 5. That's the bottleneck that we talk about. So when you say, oh my gosh, less than 5, that's really a point when if we can go from, let's say, 4 or 5 to 0, we've really, because you say, 1,000, 10, that's just, because once you get past the oocysts, then the sporozoites, you're going to get up to, let's say, 20, 30,000 again.
Correct.
So you want to, if you could somehow target the midgut when you have these oocysts, and there's just very few of them,
right,
and affect how hospitable the environment is, you can interrupt the cycle in the mosquito. You interrupt the cycle in the mosquito, you can't get to the infective sporozoite stage.
Indeed.
So this...
Yeah.
Now, the reason for working on it is a very good one.
They say there's a microbiome in the midgut, and we know that if you introduce bacteria, they will multiply. So they say, why not engineer the bacteria to produce an anti-malarial compound? And so this has been tried with E. coli, but it didn't work very well. And then the basis of this paper, they say they were looking at bacteria in the ovaries of their laboratory Anopheles stevensii mosquitoes. And it turns out they were Serratia marcescens strains.
Indeed they were.
Now, I will just stop at this time and say to everyone, I was trained many years ago to call it Serratia. Right. And I know many people call it Serratia. Dickson, how do you pronounce it?
Serratia.
Daniel?
Serratia.
There you go.
And the last part of that? Marcescens.
Marcescens, right? For sure. Right. So they sequenced it and they said, Serratia, it's in the ovary. So why don't we try feeding this to the mosquito?
Right.
By the way, this whole process of producing something in the bacteria that you then give to the mosquito that inhibits malaria is called paratransgenesis.
That's right. There's a new term.
So they did a few experiments. They found that feeding mosquitoes Serratia had no effect on lifespan, did not influence blood feeding behavior,
right,
and had no negative impact on fecundity and fertility. What do those two terms mean, fecundity and fertility?
Well, fecundity means that they can be inseminated to produce a zygote. And fertility means that they produce eggs and sperm.
Is it fecundity how many eggs they produce?
Yeah.
Okay.
Yep. But they're not...
So they said, wow, this Serratia might be a good thing to do.
Right.
So they put a gene for green fluorescent protein into the Serratia, and then they feed these to adult mosquitoes in a sugar meal. And then they say, where does it go? And they can just look by microscopy, and they can see that these bacteria efficiently populate the midgut, even if there's already an established microbiota.
Right. That's interesting, isn't it?
They go up 200-fold within a day. Yeah, because you would think that the microbiota kind of protects against colonization by something else. Yeah.
I thought that was pretty important because you're thinking, in a sense, I view this as almost a technology paper. And you want to say, do we have to first treat them all with antibiotics? Which has been happened in other sort of studies that people have looked at. First, you've got to somehow create a niche and then put them in. And what they're saying is, no, we could just put them in this sugar food, and boom, they get in there. They're gonna be able to come right into an established microbiome.
We did a paper on TWiM last week, which is not yet up, where we swallow 1.5 liters of our own saliva a day. That contains 10 to the 12th bacteria, and most of those don't colonize our gut unless you have a dysbiotic intestine. In which case, people with like Crohn's and inflammatory bowel disease, they the Klebsiella in your mouth will colonize your gut and give you big problems.
Fascinating.
So you have to have dysbiosis. But here, no, these Serratia can just go in.
Right.
They also found that these bacteria with the green fluorescent protein in hemolymph, in ovaries,
right,
and in fact, when females laid eggs, the bacteria were attached to the eggs. They could go in water and be ingested by the larvae that hatched from those eggs. So the bacteria went into the water, the larvae ate them, and they get into the offspring.
How about that?
And furthermore, here's the kicker. When they fed these bacteria with the GFP to males,
right,
they sexually transmitted them to females.
Exactly, through the accessory glands.
What's an accessory gland?
It produces seminal fluid for the male so that the sperm have liquid in order to transfer to the female.
Yeah. And these...
sort of like a insect prostate.
Yeah, I was thinking that. Yeah, that would be the human male equivalent.
That's right.
Accessory gland.
And then
That's right.
The mosquitoes that hatched from these eggs that were made from the males carrying the those mosquitoes had the bacteria in their midgut, so they persisted.
Yep.
And they did this several generations, 3 generations they tried, and it kept moving from generation to generation, these green Serratia bacteria.
Right.
So they got very excited about this. The next experiment, which is very cool, they put male mosquitoes carrying these bacteria in a cage with other bacteria that don't have the bacteria, with other mosquitoes that don't have the bacteria, to the extent of 5%. So 5% males containing GFP Serratia in a cage full of males and females with none of these bacteria. And all the offspring from that cage had the bacteria in them, 100%.
Right.
1 generation, and it persisted. This is remarkable.
Indeed.
So this thing is driving through the population. And it's, of course, everyone wants to modify mosquitoes so they can't transmit viruses and whatever else.
Yeah.
But if you make a modified mosquito, getting that gene to drive through the population is very hard. We've talked about that a lot on TWiP.
Yep.
And here, this bacterium naturally drives through.
Mm-hmm.
I find this really incredible.
Yeah, I would agree. That's why I actually cut this one out and brought it in.
That's why you sheared it from its backbone.
Sheared it. Yes, I did.
Now, the key experiment here is they then introduce into the Serratia 5 different genes encoding 5 different molecules that are anti-Plasmodium.
Right.
Effector genes.
Yes.
Effector genes is exactly right.
One of them is a peptide that binds to the mosquito midgut and inhibits Plasmodium invasion. You particularly like that one, didn't you, Dickson?
I did, because I'd never heard of it before, and I looked up the paper, and it's an interesting paper, and it blocks. You know, when the O. kinetus formed after the exflagellation events in the midgut. That's actually what happens next after they ingest the blood, is that these flagellae then seek out the female equivalent, the microgrametocyte, and they penetrate, and then that actually is fertilization. It then forms a diploid organism. And the diploid organism now has to go someplace. And where does it go? It goes in between the columnar cells of the gut tract and comes to lie up underneath the membrane that keeps the gut tract intact. And that's where the oocysts develop. And this peptide prevents them from going through that little...
Amazing.
That I love that. I just love that. That's fantastic. And by the way, it should be said that the female mosquito is the definitive host for this parasite because she harbors the sexual stages of the parasite.
Right.
And apparently there's no energy cost to the mosquito for harboring Plasmodium up to a point.
Right.
So that it's too bad, because if they were slower flyers or they ate less or something like that.
That's remarkable.
It is remarkable.
Do you think there's a benefit to the mosquito?
That's what I'm going to ask.
Who should we ask?
I guess we should ask some, maybe Tony James knows the answer. We've had him on our show, and perhaps he does have an answer for us.
Well, so the real key experiment is they introduce these effector molecule genes into the Serratia. They feed them to mosquitoes, and then they feed them 2 days later with a Plasmodium falciparum blood meal.
Exactly.
And they have a lovely graph showing
That's right.
Each dot on this graph is, I guess, a mosquito where they've counted the oocysts in the gut.
That's right.
And you see untreated, they have lots of oocysts. And then as you introduce each of these effectors in, it goes down and down. And of course, when you put them all in, you get the most inhibition. But there's still a lot of mosquitoes with a lot of oocysts per gut. If you notice, this is a dot.
It's not a sterile immunity, or a sterile protection, I should say. But that's what you'd really hope for, though, right? Even
you'd like to block it completely, right?
Of course you would, because each oocyst can produce, as Daniel said, about 10 to 20,000 sporozoites.
But these mosquitoes will still be able to transmit because they have
that's right.
They have oocysts in them, right?
Exactly right.
So this is interesting, but they have to play with it, I guess, to get better inhibition.
They need better effector molecules.
Now, Daniel, what do you think?
I mean, I had a couple of thoughts. So one is where you are right now. Is the technology going to work? Right? Is it effective enough? Are you going to get the sporocyte count low enough that you're going to really interrupt transmission?
Right.
And that you don't need 100%. Right? There's a critical mass.
Sure.
If you can get it below that. But the other is the whole sort of issue of this idea of, instead of getting rid of the mosquito, we're going to introduce Serratia into the environment.
Right.
And I don't know if it rings a bell, but are you guys familiar with something called Operation Sea Spray?
Yeah.
We
were talking
about that today. Everybody remembers that one.
Yeah. Tell us that, Daniel. Tell us what that is.
So that might, I think a lot of our listeners might not be aware of Operation Sea Spray. But we see Serratia marcescens clinically on something of a regular basis, and there was at one point it was secret, but back in 1950, the U.S. Navy had at that time a secret experiment, and what they did is they sprayed Serratia and a bacillus bacteria over the San Francisco Bay Area in California.
Lord.
Okay. And what they wanted to do is they wanted to monitor this. So they set up monitoring equipment in like 40 plus locations around the city. The Army, I think, was involved in this as well. I think the Army did the monitoring, the Navy did the release. So the Navy releases the pathogens offshore, and then it gets blown on shore, and they want to see what happens. Well, not much later, a couple weeks later, about a dozen people end up checking into Stanford Hospital with, at that time, what was considered a very rare bacteria that you really didn't see. One person ends up dying. The other people get better. But it becomes sort of interesting enough that it gets written up in the medical journal. And then there's a lot of wondering. But then finally, in the 70s, there were Senate hearings, and the Army disclosed these tests. And we sort of all find out about it. So it's very interesting. So I'm thinking about, so Serratia used to be something we didn't really see. It was considered a very rare, serious infection. Now we see it quite a bit. Of course, the Navy and the Army are pretty sure that release of Serratia had nothing to do with this at all. I mean, it didn't make any sense biologically, right?
Well, we should also say the reason why they released Serratia is because when you culture it, it's pink. So it stands out from all the other bacteria that you would grow. You know that it's so rare that if you see a pink colony, it's going to be from something that you did. So I understand also that they released it in the New York subway system. That's a rumor, but I don't know if you can confirm that or not.
I don't think I'm allowed to confirm that, but
you're not at liberty to release that fact.
Yeah, no, that's actually interesting. A few years back, I saw a patient with this really severe inflammatory cellulitis, and it was oozing this red liquid, sort of a current jelly liquid. And so the initial is, oh my gosh, it's bleeding. It's like, nah, actually. So we did a gram stain, and it was just this sheet of Serratia. And then when put in culture, as Dickson mentioned, it gives off this red pinkish pigment.
Exactly.
So that's one of the things I worry about is, so we're going to, the thought would be, okay, Serratia, we're going to put all the sugar with Serratia in it. We're going to spread it around Africa. All the mosquitoes are infected with Serratia. They're taking blood mills of all these people. And what they've demonstrated is Serratia can move right into an established microbiome maybe serratia is going to move into all the established microbiomes of all the people that are being bit and touched and interacting with mosquitoes. I have a little bit of concern here.
Interesting. That's a good point, Daniel. Glad you brought that up, because sometimes we can get carried away in our enthusiasm, but
That's right.
reminding us of the sea spray.
Well, maybe there's a way to disarm the bacteria so it won't do that and still be useful for this. It's, what are its virulent factors?
That's a good question. Yeah.
So you can play with it once you have the system, and maybe you can make it harmless. I don't know.
Maybe. A lot of work, though.
A lot of work.
Yeah.
And
reminds me of Jurassic Park, right? So
always
billions and billions of modified bacteria.
No, that's right.
That's a 1 in a billion chance.
Yeah.
Yes,
but
got 10.
We also covered another paper where Wolbachia was shown to be inhibitory for Plasmodium survival in mosquitoes, and that also is ovarially transmitted. That's horizontal as well as vertical. So. Maybe that offers a better hope.
That drives through the mosquito population.
It does.
However, Dickson, if the Army or Navy released billions of Wolbachia over San Francisco, would anyone get sick?
Yeah, the mosquitoes might. So couldn't you engineer the Wolbachia to produce these parasites?
Yeah, sure, you could.
As well. So maybe you could get 2 for 1 here.
Maybe.
Right?
People seem to be okay with putting Wolbachia in mosquitoes and releasing
I don't
them.
think anyone cares about it at that level. You're right. Because we don't get Wolbachia. But we could get serratia. I agree with Daniel. I think that's...
Yeah, I like Wolbachia more. I mean, the serratia kind of has a bad history. I mean, there's Operation Sea Spray. There was a release in Panama City. There was one in Key West. We've sort of done some...
We have.
I wonder who there in the Navy was approving this kind of... I guess no one was thinking back then that there would always be hyper-susceptible people that are going to get infected,
Yeah, let's read their last sentence because I think it's pretty definitive in that sense.
Go ahead.
It is important to note that eradication of malaria can only succeed by combining all available means, including insecticides, drugs, genetic modification, and possibly vaccines. So they know it's a complicated.
Is it a goal to eradicate malaria, Dickson?
Well, they would very much like to, but I don't know if anyone realistically could
possible,
think of that.
Right?
Everything's possible.
But you said the mosquito was the definitive host.
It is.
So that's hard.
The definitive host.
And that's going to be hard.
I know. You're absolutely right.
Because polio, only humans are infected.
Right.
Nobody.
But if you treated everyone, and if they were not drug resistant, then there would be no more malaria because there would be no way for the mosquitoes to acquire it.
But there would be a pool out there, so you'd have to treat long enough to get rid of that pool.
That's right.
Mosquitoes only live 2 weeks, so
that's right.
it's possible.
If that would be a remarkable public health program to institute. But the problem is that it's in such remote areas. I know Daniel will say, well, it's impossible because you can't get to those areas and time your drugs, even if you had them, to coincide with when you're, like, for instance, getting rid of yaws or even smallpox vaccination.
Okay.
Yeah. I mean, I may be more optimistic. I like to think I'm optimistic, but, malaria was endemic through the U.S., and it's not here anymore. Where I was in Panama, right? We'll use that as an example of a remote northwest of the canal. You do not see malaria, but east of the canal, you start to get back into malaria zones. So even, in, we'll say, an industrialized country, which the U.S. likes to believe it is, and even some areas less industrialized with the right leadership and resources brought to bear, I think one day we will get rid of malaria, but I'm just not sure how far off that one day is.
Yeah, we keep trying to reintroduce it into this kind. We have the vector, we just don't have the malaria. But sometimes we have autochthonous cases in the United States, but it's not. We have good medical surveillance and vigilance, and when someone gets sick from some unknown disease, they go to a hospital, they get treated, they get diagnosed, and you snuff it out, basically, right? But in California, I mean, due to the summer migration of migrant workers from Mexico, Vivax malaria is a real deal out there. So they have lots of autochthonous malaria. And particularly this year because of the high temperatures. I just read something about something unrelated to this topic, but vector-borne also, which is West Nile virus. they had an extended season out there. I don't know not everybody's a sports fan, but the World Series is going on now. The first game of the World Series in Los Angeles was played in 103-degree weather. That's really inhuman. Wow. They should have had more home runs than they did that game. But the point is that it was just such a hot heat wave that they were having, and it was accompanied by these fires and everything else. When you look at the environmental disturbance that's going on around the world, it's a miracle that we're still here.
All right, let's have Daniel now get back to work. He had a week off.
A new case study.
Regale us.
New case study.
Please.
Now, I think this will be a good case. We were recording some of the videos, so there were a few of the cases that I have, as we say, sort of in my can that I'm going to be presenting today and in future episodes. So, but this is a one where even though you sort of know all the information, I think it isn't a giveaway. All right, if people are ready, here's a new clinical vignette. 59-year-old Spanish-speaking female who's living on Long Island but is originally from Guatemala comes into a local hospital. This is a case I saw recently, just a few weeks ago. She initially came into the emergency room after returning from a 10-day trip to visit friends and relatives in Guatemala and El Salvador. And what brings her in is a fever to 103 degrees. She also has a cough productive of yellow sputum. She has diffuse muscle aches, fatigue, chills with fevers. And one of the first things they do is they do a respiratory pathogen panel. We used to call them viral panels, but now they're pathogen panels because they also include things other than viruses, a bunch of bacteria, etc. And this is positive for rhinovirus. They notice her procalcitonin level is less than 0.05, and they say, you know what? You're fine. It's just a virus. Go home. You'll be fine. But she comes back about 5 days later. She's not feeling well. Still fever. Still chills. Pain in her upper belly. No diarrhea, but actually she's saying she feels constipated now. Okay. And at this point, they say, we should admit you. You're still not doing well. She has no past medical or surgical history. She says she's healthy. She's not allergic to any medicines. She says people in her family are healthy. No significant family history. She doesn't take any medicine. She works cleaning houses. But a little bit more detail about her travel and geographical history. I really enjoyed getting this history from her. She says that she had spent these 10 days in Guatemala, El Salvador. Most of her time was in and around the big cities, so Guatemala City and San Salvador. She reports lots of exposure to animals, dogs, cats, livestock. But then she does offer up that she made a point of eating all the local fare, which she doesn't eat up here. She ate conch, ceviche. One of the family members had fresh eggs, so she was involved with eating the fresh eggs. And there's a flattened chicken dish that they eat that she really enjoyed while she was down there. When she's initially seen, as mentioned, she has been having these fevers. She has an elevated white count, and it's left shifted, right? So the neutrophils are increased, the eosinophils are cleared, and this is why they get me involved. She is growing Salmonella bacteria from her blood. Okay. So I get involved at this point. This is not a parasite, so it's okay. We're still going for it.
Not one of our parasites.
Not one of our. It's not one of our.
Not member of the family.
TWiP bugs. But we go ahead, initially IV antibiotics. She gets better. She gets discharged on oral antibiotics. She's just about to go out the door, and her stool oviparasite exam comes back. And it is reported. I'm gonna. This is what people have to tackle with. What do we do with her stool O and P? Rare Entamoeba coli cysts are observed. Endolimax nanotrophozoites are observed. And rare Blastocystis hominis cysts are observed.
Give me those three again.
Okay. So Entamoeba coli, Entamoeba,
Right.
For our listeners jotting these down. Endolimax nana. So that's ENDOLIMAX nana.
Nana. Okay.
Right.
Nana.
Tiny.
Okay. Tiny. This is the dwarf tapeworm, right? Do I have that right?
No, not tapeworm. Not
What is that?
It's the dwarf amoeba in this case. Endolimax.
And then rare Blastocystis hominis. BLASTOCYSTIS. Hominis.
Wow.
Right.
All three.
She has a plethora.
Have you ever seen all three at once, Dickson?
Many times. Many times.
In sick people?
Not necessarily, but
In healthy people?
sometimes.
Wow.
These are the commensals that we would talk about mainly. And we had a program on this before where we talked about the potential pathogenic nature of Blastocystis, and perhaps with so many genotypes out there are only a few that actually cause disease. But in this case, this I know why Daniel's doing this, and this is a great case for that particular thing.
Last time we decided there's no Blastocystis hominis. There's just Blastocystis.
Yeah, that's
right?
exactly right. That's exactly correct. That's exactly right.
Go ahead.
So maybe we should just to help, and I kind of tried to give a little bit of a hint, but just to make sure everyone can say it. So these are what type of organisms are these, Dickson?
Well, they're all the eukaryotes. They're eukaryotes. Okay, so they're intestinal.
I just want to know what type of organisms.
Are they amoeba?
Oh, they're all protozoans.
Protozoans.
They're all protozoans.
Yeah. So that's what I was hoping we would just let people know. So
that's
these
fine.
are
that's absolutely fine.
intestinal protozoans.
Intestinal protozoans. That's right.
And then the big question is, what do we do? Here's this woman. We're just about to let her leave. She says she's feeling better, but she's got these three things in her stool.
So she was treated for Salmonella with an antibiotic, I presume.
Yes,
that's right, like ciprofloxacin or something like that.
Yeah, she was initially treated with IV antibiotics, but then she was discharged. Now this is from when she was admitted. So this comes back from the initial admission. ONP.
But it took the data long enough to reach her that she was almost discharged by the time it got there.
By the time, yeah.
They said, "Oh, wait a minute here.
Wait a minute, Dr. Griffin. What do we do with this?"
But she feels pretty good, right?
She's feeling great.
Her diarrhea's probably gone away.
No more diarrhea.
She feels better. She feels great. And I'll tell people too, I'm going to see her back in clinic. So I see her back in clinic 2 weeks later.
Good point.
She says, "Dr. Griffin, I feel great.
Yeah.
No more symptoms.
Right.
But what is up? What are we going to do about this?"
You let her go home then. That's what you're saying.
I will say that too. Despite these results, I let her go home. We'll talk about whether or not I did anything as far as treatment in addition to the Cipro, but I let her go home. I felt like I could manage this patient outside of the hospital, the acute hospital setting. I see her 2 weeks in clinic, and at that point, she's doing very well. Right. And so for our listeners, it's going to be a question of what did I do that made me feel comfortable sending her out? And then what did I do in 2 weeks when I see her in clinic?
Bingo.
It's good. I like it.
I do too.
A little twist.
A little twist.
Another twist is parasitic. Oh, no, that's something else.
Another twist is licorice.
This is very good. I like this case. I like it because it has a nice virus in it.
Oh, yeah.
We have virus, we have bacteria, we have parasites. We got all the stuff.
The works.
Okay, very good. Go for it, folks.
Yep.
Let's hear your guesses.
That's right.
Do a couple of email here, and then we'll wrap it up.
Well, I want to say something also about the listener responses. We've got these textbooks sitting doing nothing. Shouldn't we give one away this time?
This one, the time we just did now?
What do you think? We have it in our power to give away a hard copy of Parasitic Diseases, 6th edition.
We had 1, 2, 3, 4, 5.
I'm wondering how we should do it. Why don't we say for this one that we just presented, if you get it the first one or the 7th one, what do you want to pick?
Well, I had a different number. I had a different number. That's right.
What were you thinking?
Vincent,
give them everybody.
Yeah. Well, what if we get 20 responses?
All right, we had 8 responses.
Then,
so you want the next one, or do you want the one we just did?
I think in the future, so we have. You want to give those out?
The lady with the 59-year-old lady, or the
one that Daniel just gave as a number.
So next time we'll
award.
Right, we're gonna whoever's right, we'll take a random number,
A random number.
Yeah.
Right. So we take 1 through 8, randomize the numbers, and pick it from a hat.
Exactly.
Whoever gets that number
Gets the book.
they get to go home with the door prize.
You get a copy of Parasitic Diseases 6th edition hard copy.
Right. And one of us, or maybe both of us, will sign that book.
Yeah, you
So
should.
we'll have a signed copy.
Okay.
Maybe.
Maybe. All right. Let's read a couple of emails so we can just do a few here.
Absolutely.
Hannah writes, Dear TWiP hosts and associated parasites, I just came across this blog post from an entomologist acquaintance of mine and thought it would be of interest. It chronicles the author's experience with several human botflies, details their biology, and features some gorgeous photos of one of the larvae, even in situ, as well as the adult fly that it developed into. She gives a link to that. Keep up the good work. And, of course, Hannah also submitted a guess.
Right.
So that's timely.
Indeed.
All right. Now, Dickson, can you take the next one? It's rather long.
I will do my best. Anthony writes, Terry the earthworm takes a wrong turn at the tree stump in TWiP 52. And he gives a link to that episode. There's a bit of discussion on how might earthworms happen to be cavorting underwater. I guess we made a reference to that. Earthworms that are washed into a pond or lake or river are most likely to be quickly consumed by fish, salamanders, frogs, or turtles. Earthworms won't consume parasite larvae in the water. Earthworms will drown. I think that the reference mention of aquatic oligochaetes meant just that and not an earthworm that made a wrong turn at the tree stump. And gives another reference to that.
You know, this is 52. That's a long time ago.
It's a while back. So it's, it's past history here. What are you doing to us? Tubifex, and then he lists that, and blackworms are commonly used in aquaculture, tropical fish in particular. It's long been thought that fish catch disease from eating tubifex. This is absolutely true, by the way, including conditions related to parasites, and I'll tell you one when we get to the answer. Blackworms are considered cleaner. I don't know if that belief is based on fact or faith. I wonder if aquatic oligochaetes transmit parasites or other diseases, and indeed they do, in nature or under domestication to economically important fish, and indeed they do. Robert J. Goldstein is a parasitologist and a prolific writer on exotic fish, and he gives another link to that. This appears to be him on LinkedIn, and he gives a link to that. I don't recall reading any discussions by Dr. Goldstein on parasites of fish from tubifex or blackworms. A friend is in communication with him and could provide contact information. Thank you. Tropical fish expert Mark Weiss tells me that Dr. Goldstein has some obscure fish parasite named after him. By the way, on a separate note, I shuddered when Dr. Griffin conflated the founder of the field of tropical medicine with the American horror. I must admit, though, that the mnemonic was remarkably effective. Glancing at this Facebook post, then he gives a link to that. Immediately, I recognized S. Manson-I after never having seen a picture, but only having heard Dr. Griffin's description. So, in response to your question, do tubifex worms transmit diseases to fish? The answer is yes. They transmit something called whirling disease. Mm-hmm. And whirling disease is a protozoan that was imported from Europe through aquaculture of trout. And indeed, the tubifex worms appear to be important as an intermediate host for this parasite's life cycle. So it's a very interesting life cycle indeed. And the only way to get rid of it is to allow the host definitive species, in this case the trout, to become resistant by attrition. And that's basically what's happened in nature, is that the parasite was introduced. It particularly attacks rainbow trout because those are native to North America, and this is a European-based parasite. The brown trout, which was introduced from Europe to the United States, is not as susceptible to this parasite because it has evolved resistance through, as I said, natural selective pressures by the parasite, killing off the ones that couldn't resist, leaving the ones that could. Right. And that has actually happened in this country. The rainbow trout populations in the western rivers that have had experience with the Myxobolus cerebralis, which is the protozoan parasite, have become resistant to infection, and the tubifex worms, which make up a part of their diet when they're small, maybe an inch-long fish, no longer are a threat to their existence. And of course, in rivers that haven't had this experience yet, it's still a threat. So whirling disease still exists, but it's not as prevalent as it used to be. That's a great set of questions and answers, I think. I'm glad to participate in it. If anybody has anything else to add out there, please join in.
Daniel, what's this conflating the founder of the field of tropical medicine?
Well, you referred to a certain nefarious person who was, I think, didn't you, like Patrick Manson?
To blend, fuse, unite, integrate. So what I did is as I did the mnemonic with thinking about which vectors cause which diseases, often on tests you might have to distinguish different schistosome eggs. And how do you tell which are the schistosome hematobium, Mansoni, or Japonicum? And the mnemonic that I suggest there is the Charles Manson egg.
Yeah, exactly.
Have this big, huge spine sticking out of the side. It's actually Patrick Manson, one of the founder, if not maybe the founder of the field of tropical medicine, parasitology. Quite a superstar of parasitology. Yeah. But so these are mnemonics. Now, the hematobium, which is the one that's going to involve the bladder, the urinary system, that actually has the spine right at the end, so it allows it to pass out the urethra, pass through the urinary system. And then the Japonicum is so refined that it's actually even very hard to see the spine. It's just symmetrical. It's so making people think of the refined Japanese culture.
Oh, wonderful.
So it's one of my mnemonics.
Charles Manson, Patrick Manson.
They were not related to mine.
That is the American horror.
Exactly.
Daniel, can you take John's?
Well, he does write Hi Vincent, but Hi Vincent. Hope all is well. Let's go out to recent victims of the Mexican earthquake and Hurricanes Harvey and Irma. I thought the following might be an interesting discussion for TWiP. GSK completed a clinical trial on Moscarex in 2014, indicating a low efficacy. Nevertheless, an efficacy that would have a significant impact on malarial disease in sub-Saharan Africa if vaccination was conducted in mass. The company is part of a multilateral partnership that includes funding from multiple sources, including the Wellcome Trust and the Bill and Melinda Gates Foundation. And he gives us a link here. A recent study conducted by Professor Karen Day et al. In Gabon has contributed to validation of the strain theory developed by Professor Sunetra Gupta and Professor Day. Surface antigen variation could threaten efficacy of a malarial vaccine. And then we have some links here. It would be interesting to hear what insights the TWiP panel could bring to the fore on challenges that face malaria vaccines. Apologies if this has been covered in a previous episode. I haven't caught up yet. And thank you again for all you do. Best regards, John D.
Do you know anything about this variation business, Dickson?
I don't, but I do know that in the beginning of exploration of this parasite's biology, they did find that it had variant antigens, which they could, link to the concept of antigenic variation.
Yeah.
But it didn't involve the same mechanism that trypanosomes use. But Giardia has antigens that vary as well. So, I mean, in fact...
But for a malaria... This vaccine, by the way, induces responses to the circumsporozoite protein.
Right.
Does that undergo antigenic variation?
Not that we know of. It's a pretty constant quadrapeptide that's repeated again and again and again.
But is that a good vaccine antigen?
They've tried this until they're blue in the face, and it reduces the infection, but it doesn't work. But the data say that it should, because when you take irradiated sporozoites, which can't go beyond that stage, and they're still, quotes unquote, alive, you can induce a sterile immunity with them. So that stage has the key to understanding immunity to malaria, but they haven't cracked it open fully yet.
Of course, there are other kinds of vaccines being developed as well, right?
You bet. Oh, yeah.
All sorts.
They've made them. And in fact, the paper that we reviewed just recently, today, for instance, there's a vaccine against the developing stages in the midgut of the mosquito that you can immunize a mammalian host against, so that when the blood is swallowed, then the antibodies are there in the serum portion of the blood that they took the blood meal from. When the ookinete is complete, it kills them. So that's called an altruistic vaccine. Although it doesn't protect the person that has the antibodies against the parasite, it prevents the spread of the parasite to the next host. So,
yeah, no. So this vaccine was Well, the whole vaccine concept has been a challenge in malaria for, decades. And this was, you had to do a series of 3 shots. You had to do them, if I remember correctly, every year.
That's correct.
And you were getting a significant reduction in the number of children, particularly, that would end up hospitalized with malaria. I think it was about 50%. And so there was a lot of sort of buzz, excitement. But is that enough considering the resources? And so we're really hoping for something that works at the level of vaccine sort of standards that we're used to, 80, 90%.
Exactly.
Because there is the concern that, you do this for a while, you put all the money and resources in, you're only getting about a 50% reduction. Which is a lot, actually, when you think about the number of kids that get hospitalized and sick and actually die.
True.
So
a lot of the effects of some of the other vaccines were to prevent death, which is, of course, a great endpoint to use as the result of having a vaccine at all. But there is some loss of cognitive function with each succeeding cerebral event that you get from malaria, so that even though the vaccine would prevent you from dying, it wouldn't prevent you from suffering.
Right. All right. One more. Chris's email is about the case from TWiP 139. This is the woman with persistent eosinophilia, Daniel.
Mm-hmm.
What was the solution to that? Remind me.
Yeah. Do you guys remember? I can look up here.
I don't remember.
Do you want to? You read it, and I will.
Well, I don't want to read it. I'm not going to read the whole thing because it's late. And
no,
back
actually,
to that.
I have a list here, so I'm opening it right now to see what the case was. 139. Oh, yes, this is the woman who went to where amazing things happen, right? And then I think I remember
yes.
Right. So she was in the Peace Corps, and then she came back, and she had Loa.
Oh, Loa. Okay,
that's the one. That was the one.
So he gets strongyloides wuchereria,
right,
but not Loa.
No.
The last email is from Anthony, who writes TWiP 59 errors. So Anthony must be going back to all the old.
Anthony, leave us alone.
He writes,
this is before I joined, so that's my disclaimer.
He writes, canaries do indeed come from the Canary Islands.
Uh-huh.
I understand that they continue to do so. I've read that canary breeders on the Iberian Peninsula mate wild birds with domestic stock to add variety to the song. The birds are named after the islands, not the other way around. The ever-practical Romans were impressed by a large breed of dog bred there, not little singing birds. They called the place the Dog Islands.
Oh.
Canary is derived from Latin for dog, as in cave canum.
Interesting.
Birds are not divided into passeriforms and waterfowl. Eagles are not passeriforms. If I actually heard that. That was probably Dickson.
I didn't say that.
On a separate but related note, and this time it turns out to be a pun, I'd have to look up the citation, but canary song is negatively affected by parasite infection. That could mean that singing gives the hen a clear indication of the health of a potential male. Yeah, it could. Sure.
That's interesting.
Made total sense.
And by the way, did you know that pencils come from Pennsylvania? That's
Anthony,
actually a
don't
line
listen
from a
to
song.
Dixon. You can keep it up.
It's a line from a song. Don't worry about it.
That's TWiP 141, gentlemen.
Yes.
You can find it at Apple Podcasts and microbe.tv/TWiP. You can send us your questions and comments, twip@microbe.tv. And if you're interested in giving us some financial support, microbe.tv/contribute. Dickson Despommier is at Racaniello.org, livingriver.org, and parasiteswithoutborders.com, right?
Right.
Thank you, Dickson.
You're welcome, Vincent.
Do you need a ride home?
I do.
No problem. Daniel Griffin is at Columbia University Medical College, parasiteswithoutborders.com. Thank you, Daniel.
I think it's center. I think it's.
What did I say?
College.
Columbia University Medical Center.
A pleasure as always.
I'm glad he's listening.
Yes.
I'm Vincent Racaniello. You can find me at virology.ws. I just realized I bought virology.blog also.
Nice.
Isn't that cool?
It is
So
cool.
you can go to either one. Ends
That's
up
good.
at the same thing.
Very good.
And I want to thank the American Society for Microbiology for their support of TWiP, and Ronald Jenkins for the cool music we use.
You bet.
RonaldJenkins.com. You've been listening to This Week in Parasitism. Thanks for joining us. We'll be back soon. Another TWiP.
is parasitic.